Greetings everyone and welcome to my talk. My name is Fabio Calcinelli, I work at the Graz University of Technology, and over the next minutes I will tell you about the thin films of organic molecules and about the way in which different substrates can influence their geometry. To get started, we all know that all properties of a material depend on its structure. This is particularly true and relevant in the case of organic thin films, which are a class of materials with a wide range of applications. For example, one of them is organic electronics, in which their flexible nature allows them to be used in wearable devices or foldable screens, but also pharmaceutical is another, and there's much more. And for all these applications, the relevant properties depend not only on the species of organic molecule that is used, but also in the way in which all the molecules are arranged in space. And in turn, the way in which the molecules are arranged in space can widely vary and depends on several factors. One of them, the one we will focus on today, is the substrate on which the thin film is grown. So, in synthesis, the question I would like to answer today is how can different substrates induce different polymorphs for a thin film, and also whether this difference will always manifest straight away from the first layer or not. The paradigm we're adopting in this is that we assume that the structure that will form will be the most energetically stable one. Of course, we cannot calculate the energy of all possible polymorphs because their number leads to infinity and we don't have infinite computational resources, but luckily there are ways to get around this problem. What I'm referring to in particular is the sample approach. SAMPLE stands for Surface Adsorbate Polymorph Prediction with Little Effort, and it's a software package that uses the results of a few DFT calculations, combines them with some machine learning, and attempts to answer one specific question, which is, given one substrate and one species of organic molecule, which configuration, which polymorph will the first layer of molecules assume? So, in order to apply the sample approach towards thin film prediction, we have to start looking at the second layer of adsorbates. And to do this, we have to take at least two fundamental assumptions. The first one is that the Frank van der Merwe growth mechanism is at play. What this means is that only after the first layer is completely formed, only then will the second layer start to form. Also, we have to take the assumption that as consecutive layers form, the first layer will remain perfectly fixed. So with these two assumptions in mind, we can start looking at the systems we want to study. One of them, as shown here, is a geometry for the first layer of benzoquinone on graphene, and the other is a geometry for the first layer of benzoquinone on silver 111, as obtained in a previous work of our research group. What is interesting here is that we see that two structures that are basically identical once we account for different unit cells for each of the two substrates, while these two basically identical structures appear for both substrates at extremely favorable rankings in energy. And what this means is that we can look at a situation in which apparently different substrates may seem to induce the same geometry for the first layer. So with the sample approach, we can now take these two structures as substrate and predict all polymorphs for the second layer, then rank them and take the best ones. This is what we do here. What we see is in black molecules of the first layer, in color molecules in the second layer, while the substrate atoms are simply not shown for simplicity. The most interesting thing we see is that we have two structures that return basically unchanged for both substrates. One is molecule on molecule, in which each molecule of the second layer is centered on one oxygens of molecules in the first layer, and the other is molecule on gap, in which the molecules of the second layer are centered on the gaps between molecules of the first layer. And the most interesting thing is that these two structures return basically unchanged, but now their ordering is switched. In fact, in the case of silver 111, molecule on molecule is the most stable, while in case of graphene it's molecule on gap that prevails. So the first thing we have to ask ourselves if this is due to a direct effect of the substrate, or rather to some small, apparently invisible differences in geometry between the two different substrates. What we do to ascertain this is shown in this plot. What we have is the absorption energy of the two configurations, and we see how this absorption energy varies when we remove the substrate atoms from the unit cell. What we observe is that in the case of graphene, the result of this is a destabilization of both configurations, and a destabilization by roughly the same amount. And this can easily make intuitive sense, because by removing the carbon atoms of graphene, we are removing a source of a purely attractive van der Waals interaction. On the other hand, in the case of silver, we see that not only the destabilization is larger, but is also relevantly different for the two different configurations. So it would seem that there is something in the way silver atoms interact with adsorbates that produces a strong stabilization, but also a stabilization that's different for different structures. On the way to understand what this interaction may be, we can start by considering that silver 111 is well known to be an electron-donating substrate, and what we can do is to look at the amount of charge that's transferred from the substrate to the adsorbates. If we do it, we can see that this is rather irrelevant in the case of graphene, but is instead quite important in the case of silver 111, for which it amounts to about 0.25 electrons for each molecule in the first layer. What this means, basically, is that in the case of graphene, the lowest unoccupied molecular orbital of benzoquinone will remain empty, but in the case of silver 111, it will get filled by around 0.25 electrons for each molecule. Now, we have seen what makes the two substrates different. Now let's see what makes the two configurations different. And to do this, we look at a very simple model, which is a molecular dimer. Two benzoquinone molecules, one on top of the other. We then take the top molecule and we shift its position along the longer molecular axis. And in doing so, we pass through the positions that correspond to molecule on molecule and to molecule on gap. Then we evaluate the energy of the system in each position, and what we see is that molecule on molecule appears to be less stable than molecule on gap. What we can do now is to repeat the procedure, but this time adding extra electronic charge to the system. What happens then is that the profile of this energy curve changes, that molecule on molecule and molecule on gap become two local minima, and that molecule on molecule goes from being less stable than molecule on gap to being roughly as stable as molecule on gap. So, apparently there is something in molecule on molecule that makes it better compared to molecule on gap at hosting additional electronic charge. We can understand what this factor is by looking at the overlap between the lumos of the two molecules. In fact, we see that the LUMO overlap for molecule on molecule is greater than for molecule on gap, and that in general there is a very nice correspondence between LUMO overlap and stability in the case of the most charged systems. This makes intuitive sense because we know that if two orbitals of neighboring molecules overlap, then the stabilization of the resulting orbital depends on the intensity of the overlap. So if we have electronic charge on one of the LUMOs, the higher the overlap is with the other orbital of the other molecule, the more delocalized the charge will be, the more stable the system will become. Of course, this only plays a role if there actually is some electronic charge in this LUMO, if it is not actually a completely unoccupied molecular orbital. So, to sum up, we have seen that different substrates can give different charge transfer, and we've also seen that different configurations can offer different LUMO-LUMO overlap between different layers. And this LUMO-LUMO overlap is a stabilizing factor in case there is the presence of an additional electronic charge. So we can see from the interplay of these two factors, we have that different substrates stabilize different polymorphs. In so doing, they induce different structures, and they do so in a way that only becomes apparent after the first layer is completely formed. To conclude, I would like to thank my research group, my supervisor Oliver Hoffman, our collaborators from the Technical University of Munich, Harald Oberhofer and Simeon Kahn, and finally thank funding agency, the computational resources I've used, and lastly, but not least, all of you for your attention.